Okay, so welcome Alice Millbank to Future of Foods interviews. And you are a PhD researcher working on cultivated meat bioprocessing at Aston University. And you have a focus on scaffolds, particularly mycelium scaffolds for growing cultivated meat. So can you sort of just tell us a little bit about your work there at Aston and what the problem is that you're trying to solve?
Yeah, thanks, Alex. So my PhD is all about how we scale up the production of this novel food technology. So most cultivated meat starts with animal stem cells, so the cells that the animal relies on for repair and regeneration. So when their tissue is damaged, for example, these cells are activated, they multiply, and then they transform into the specific cell types that need replacing. So in cultivated meat, we harness that natural process, but in a clean and controlled environment to grow real animal tissue without the animal. But to grow these cells outside of the warm and nourishing environment of the animal's body, we need to recreate some of the essential conditions. So things like nutrients, oxygen, and importantly for many stem cells, something that they can cling on to. So that's where my PhD project on scaffolds comes in. So a scaffold is a supportive structure that cells can attach to and grow on. But the challenge is that since cells need a surface, surface area then becomes a limiting factor. So you quickly run out of space, which makes scaling incredibly difficult. So that's one of the major bottlenecks in cultivated meat bioprocessing. How do we grow adherent cells at a truly large scale? So my PhD aims to tackle that by exploring new scaffold materials such as mycelium that could unlock much higher cell yields.
Okay, and mycelium, just for people who don't know, I'm sure everybody does, is the sort of root structure of fungi, right?
Yeah, exactly.
Yeah,
Yeah. So most people know fungi as mushrooms, but those are just the fruiting body. So the main organism is what's underneath our feet. They create these vast networks of mycelia.
yeah. And so the idea of having mycelia, which is edible, as the scaffold, means, I mean, what would happen if you didn't have a scaffold? What would happen with the kind of cell growth, etc.?
I mean, it really depends on the stem cell. You can get stem cells that grow without scaffolds, but the purpose of a scaffold, well, it really depends on the kind of meat that you want to grow. So if we're looking at whole cut products like a steak and a chicken fillet, then we need larger structured scaffolds to help those cells grow in a really controlled manner. Um, but this is really technologically difficult to achieve and not a scalable technology as of yet. At the moment, my focus is on unstructured products, so things like mince and burgers and sausages. And what scaffolds allow us to do in that regard is to grow the stem cells at scale. So this is because we usually grow unstructured meat in bioreactors, which are essentially the same kinds of tanks that we use for fermenting things like beer. And inside a bioreactor, the environment is really dynamic. So we stir all of the cells around in something called media, which is basically a nutrient broth. And this allows them to receive a steady supply of food and oxygen. So instead of big structured scaffolds like we'd use for whole cuts, we have to use something known as a microcarrier. So these are basically tiny scaffolds. It's tiny versions of scaffolds that are able to be suspended in the liquid. So by shrinking that scaffold down to a microscopic particle, we're really increasing the surface area to volume ratio. So that means that we can grow cells at a much higher density in the same physical space. So I'm just going to ask you to take part in a quick visualization experiment, Alex, for
Okay.
those people watching on YouTube. So if you press your hands together like this, great. So this is our basic scaffold. Okay. So stem cells can attach around the external parts of our hands, but there's very limited amount of surface area. So we'd quickly fill up a bioreactor if we filled it with scaffolds like this. So we'd end up with a product that's more scaffold than meat cells. So it's just not a very efficient use of space. So if you just separate your hands just slightly for me, Alex. Great. Okay. So what's happened?
You have increased the surface area.
Exactly. Yeah.
Yeah.
Okay. So spread your fingers apart like this.
Okay. Yeah.
We've increased surface area again. So it doesn't take up that much more space. And this is the principle of microcarriers. So by designing smaller scaffolds, we're significantly increasing the surface area available for cells to attach on.
Okay. So kind of similar in a way to the bits in our lungs. What are they called? The
Alveoli.
the alveoli. Yeah.
Yeah,
Yeah.
exactly.
And roots, I suppose, and mycelium. Yeah. So they have a large surface area. So kind of mycelia, because it grows like that, it has that, and so it would be alive, would it, in the bioreactor?
No. So I would like to try putting them in whilst they're still alive, but at the moment I am having to kill them. So um, I have to sterilize them, and I'm at the moment using something called autoclaving. So I use a high temperature and steam, and it kills them off. So I would like to experiment trying to put them in whilst they're alive, because I have a theory that they could possibly provide byproducts that would then help to feed the cells. But that's a future project. But you were exactly right. So the fungi produce long branching networks of fibers. And this is sort of a biomimicry effect, actually. And it's something that we often try and achieve when looking to design scaffolds, is to try and create something that mimics the natural environment within inside the animal's body. So fungi offer those long fibrous, lots of surface area, and they're actually more closely related to humans than they are plants. So this is one of my so this is one reason why I think that the stem cells like them so much, because they offer a surface that is more familiar for the animal cells to grow on.
Okay, so it's a case of the cells have to choose to attach to a scaffold, do they?
Yes, yeah. So it's it's uh, it's important to consider the biology of the cell behavior, as well as the design and engineering of the scaffold, because ultimately you can create something that is ideal in size, that's cheap to manufacture, that's got a great texture. But if the chemistry is not there, if the cells don't like the scaffold, they're not going to attach. So it's always this dance between what the cells want, and also what we can sustainably and ethically produce at scale.
Okay, so what sort of things would the cells reject as a scaffold?
So they so traditionally in tissue engineering, for example, we have used materials such as glass and plastic, and we've coated them with animal-derived proteins like collagen, and stem cells absolutely love collagen. So this has helped us to attach them to many plant-based materials as well. So that's a good example of something that stem cells aren't really that keen on. It's like plant-based polysaccharides. They have been used within the cultivated meat industry, but we often have to functionalize them. We have to add things to coat them with something that makes them more appealing.
Okay, so is there something that fungi and mycelium has that's kind of closer to our own biology or the kind of biology of meat that is kind of closer than plant-based scaffolds?
Yeah, so there's a paper that came out relatively recently that looked at the percentage of RGD motifs that were present on fungi. So RGD motif is basically a small amino acid sequence that stem cells like to attach to. You don't really find them very often in the plant world, but as I've mentioned, fungi are more closely related to humans. So there are a number of species that contain these RGD motifs. So, for example, cordyceps mushrooms, there's shiitake mushrooms, oysters. They all contain quite a high percentage of RGD motifs. So that's what I think. But it's at the moment it's just speculation as to why I think they're attaching to my scaffolds. We have to do some further work to work out the how and why.
So when you say that fungi is more closely connected to us than it is to plants, that's in its DNA, in its structure.
Yeah,
I
yeah,
mean,
yeah.
can you make that clear to a kind of non-science person how that actually works?
Yeah, so when during evolution, at some point we've got a more closely related ancestor. So there's different kingdoms of life. Plants have their own kingdom, fungi have their own kingdom, and humans have their own kingdom. And at some point we were more closely, we shared a more common ancestor than we did with plants.
Okay. So we have an ancestor in common. Right. Okay. Interesting. I'd like to meet that ancestor. That wouldn't that be interesting? So, so, so how did you come upon mycelium as a potential uh, solution to this problem?
Yeah, great question. I've just always been fascinated by teeny tiny things. So I studied microbiology. So I've worked on viruses like Ebola. I volunteered at a lab where we explored the different compounds that were made by soil bacteria. And more recently, I've just learned. I've been getting my nails really dirty, going on forages and learning more about the vast networks of fungi underneath our feet. And so I think I've just become infected, a bit like is it The Last of Us? Is it The Last of Us? What
I
do they call
almost mentioned that program earlier when you were talking about live mycelia in the bioreactor
it?
and kind of potential kind of consequences of that.
Yeah.
Who knows? Yeah.
Yeah, and my family and friends really do think I've been infected because I do go on about fungi quite a lot. You probably noticed my picture on the wall. I've got fungi earrings. I just think that they are fantastic, really interesting organisms. They have so much potential to help us in everyday problems. We're using them to grow leather. We're using them to break down plastics and oil spills. So I just started to do a bit of research, and I noticed as well because I mentioned something called biomimicry, and when I was looking at the project that I was going to be working on and the different requirements of a scaffold, one of the things that is quite essential is that the scaffold replicates the living environment inside the animal. And I just thought about things that replicate that in nature, because you also have to consider the fact that it needs to be edible. And I know that fungi are, some of them are edible. So it got me thinking about whether or not we could grow them on mycelia. And it was just a wild, it was just a whim, to be honest. I just thought, oh, I wonder if they'll grow, because I was previously working on a different material, but I persuaded my supervisor to let me try this new material. And I went to visit a company called Myconius, and they showed me the different strains of fungi, including some that grow naturally into these small little spherical tangles of fibers. And I just thought, wow, these are basically naturally made microcarriers. They were just perfect. They were small enough. They had lots of surface area. They were fibrous. So yeah, I gave it a go, and the stem cells seemed to really like them.
Yeah. Your supervisor is Irene, right?
Yeah, correct.
We should give her a little mention as well.
Yeah, definitely. She's been fantastic, and has encouraged me to follow my interests, and has very much just allowed me to do what I want to do. Um, yeah, she's been great.
Yeah. And so are you the only person, would you say, or one of the few people working on this technology or on this research? Because I'm sure I heard a couple of years ago that this was a potential scaffold. So is that from your research that I would?
So I just thought about it without having come across it, but after I started working on it, I found that there were various different groups that have already tried growing on fungi. So there are some groups that work on so mycelia can grow in a liquid broth, or they can grow on a solid substrate. Um, so you mentioned to me before, Alex, that you've grown mushrooms at home. That's most likely on a solid substrate, right? So
Yeah,
grains and coffee grounds and that kind of thing.
yeah,
So when you grow it like that, you get a much more dense, like solid chunk of mycelia, and that's when you can grow something like a whole cut, that you can use as a whole cut scaffold. Um, and I know there's a bunch of groups working on that, but in terms of the small spherical microcarriers, there, as far as I'm aware, there's just one other group that's working on this at UC Davis. They're using different strains, but yeah, and different stem cells as well. So I'm using stem cells that come from cows, bovine adipose derived, and I think they were using fibroblasts, um, and C2C12s, which are a type of mouse cell. So yeah, but it's exciting because
okay,
people are starting to realize their potential. And there was one other study as well that autoclaved mushrooms, just mushrooms that you buy in your supermarket, like shiitake mushrooms and oysters, and they autoclaved them,
what does
and then,
autoclave mean?
so it's just where you use the high temperature and steam to sterilize, and it can also kill various organisms. So yeah, so they just sterilized it and then added the stem cells directly to the mushrooms. So
Okay.
that's an alternative way of
So
growing.
how are you growing your mycelia so that you get the kind of long threads of mycelia? I mean, I'm assuming that's what you put into the bioreactor, the actual threads, right? The kind of long.
Yeah, so they're microscopic. Well, some of them are bigger. They range in size depending on which strain and which process you use to grow them.
Hmm.
But it all starts with spores. So you take spores. I use spores that are used within the dairy industry. So myconios, they work with different strains of fungi that can be used to ferment cheese. So blue cheese, camembert. And then I take the spores that they provide. I grow them up by um, I I add them to some media, so the nutrient broth, um, and then I shake them gently um, at a set temperature, and they start to germinate. So they turn into these long fibers known as hyphae. And because of the gentle rocking motion, they just sort of gather together. They tangle up, and
Okay.
then they form these little hairy spheres. And yeah, you just get this big flask full of tiny little. They look spherical, but when you look at them under the microscope, you can just see that they're just tangled up fibers.
Wonderful. And what's the media you're growing them in then? How, yeah, what's that
Yeah,
exactly?
they're not fussy at all. So at the moment, I use a potato broth. So it's just yeah, you can pretty much use anything. You just blend up potatoes, and yeah, at the moment myconios provide me with the media that they use, so I'm not aware of all of the ingredients, but I know that you can train fungi to grow on using different nutrients, and that's actually one of the things that is quite exciting, because they grow on so much. You could use waste products from different industries. So at the moment, I've got some oyster mushrooms that I'm growing on used coffee grounds from my local cafe. And there's a scientist called Peter McCoy. He's trained his oyster mushrooms to grow on used cigarette butts, which is not something you'd want to eat, but it just demonstrates that that you can harness fungi as part of a circular economy.
How interesting. Yes, and just to mention that I have had myconios on a podcast if anyone's curious to know more about their cheese making. Um, but yeah, that is fascinating. So once you've grown them in the media, um, what do you then do to use them as a scaffold? What's the next process?
Yeah, so they are just really easy to use as microcarriers because I don't really do much. Um, I just autoclave them and then I soak them in media for cell culture, for stem cell culture, and then I just add the stem cells, and that is pretty much it.
Okay. And that's in a bioreactor, is it at that point? And that's kind of that would be sort of shaken still that you would kind of carry on moving that with kind of bubbles that you'd put through, would you? How would
Yes.
yeah.
So the first year of my PhD, I grew them in static culture. So that was just in these tiny little well plates, and that's just to gauge whether or not they're going to grow, and to optimize the conditions. I've started to move them into spinner flasks, which are small bioreactors, and at the moment, I'm working on optimizing the bioprocess to grow them in a more dynamic environment. My There's a team at Aston who are working on trying to um, re-engineer the spinner flask to make it more translatable to the larger bioreactors. Because
Hmm.
at the moment, spinner flasks use a magnetic impeller. So there's a long glass tube, and on the end there's a sort of paddle, and there's a magnet that you put this spinner flask on a platform, and then the magnetism, it spins the impeller around. So
Okay.
um, the problem with that is that it's not engineered to be precise. So every time you use this spinner flask, you can put the impeller at different depths. You have to do it by eye, really, and that will change the way that the liquid inside the spinner flask spins. And the impeller as well, it doesn't match the shape of the larger impellers. So everything we're trying to do is we're trying to make sure that when we're starting to scale up, that the optimization work that we do at a smaller scale is going to translate to the industrial size scales. So yeah, we're working on 3D printing our own impellers, using a motor instead of a magnet, so that we don't have that problem with things get stuck in the gap that the magnet goes through, and yeah. So that's, yeah, watch this space because we're
Okay.
working on that as we speak.
It sounds interesting that the potential is huge, and you can kind of tweak things here and there and kind of make a big difference. So, I mean, you mentioned that the mycelium isn't fussy. So could you grow mycelium in the same serum as the cells so that they could all be growing at the same time and just sort of finding each other?
Excellent question, Alex, and one that I want to test. At the moment, I don't know, but it would be very interesting to find out. They do grow at different temperatures. So the mycelia that I work with, they grow best between 25 and 27 degrees. So when you grow stem cells, they grow at a balmy 37 degrees because that's the temperature that you have in your body. So I would be unsure whether they would grow very well at that temperature.
But the
But
mycelia is kind of less fussy, perhaps. So
yes, yeah,
it could.
yeah. And what I'm hoping is that they'll, if we do try growing them together as a co-culture, so keeping the mycelia alive, then I'm hoping that they'll still be able to maintain staying alive, but it won't prioritize their growth. It won't they won't compete or outcompete the stem cells,
Right.
because what we're really trying to do is to proliferate, so to grow and multiply those stem cells as much as possible. Yeah. So, but this is something I've not experimented with yet. I'd have to focus on a future project for that.
Yeah, fascinating stuff, isn't it? So, how much does sort of, I mean, are there any legal constraints around the work that you do, sort of regulatory alignment, food grade safety, and that sort of thing?
Yeah, yeah. So these are really important to think about. So there's no point in designing an edible food product that is not safe to eat, and it's not enjoyable for people. So if people can't or won't eat it, it just defeats the whole object of cultivated meat production. So I think food grade thinking has to shape every decision from day one. At Aston, we're going one step further. So we closely work with our psychology team, and they study consumer acceptance of novel food technologies. So it's one thing for people to say, "Oh yes, I'd try cultivated meat," but behavior doesn't always match what people report. So Dr. Jason Thomas and his team, they use psychobiological approaches. So things like brain activation patterns and salivation sensors, to get a more honest read of how people really respond to novel foods. So it's almost like a scientific lie detector. Um, we strongly believe at Aston that consumer trust shouldn't come after the product launches. So it should actually help us to inform the design process from the very beginning.
Hmm. So, I mean, that's assuming that they are faced with the product in front of them and it's on a plate and that they can kind of taste it. But I suppose before it gets to that point, it's all about the kind of coloration and the information that they're given and the kind of packaging and the naming and all of that stuff to actually make them go and buy it from the supermarket.
Yeah, but we are allowed to give them a plant-based burger, for example, as part of a study and pretend that it's cultivated meat. So you
Ah.
can get people thinking and measure salivation and things that way. But yes, that it's also important to do surveys and to gauge people's understanding and their perception of different materials that we might want to try.
Have you tried that experiment then with plant-based meats? No.
Not yet. No, no,
Okay.
no. I know this is something that Jason really wants to work on.
So you tell people it's cultivated meat and kind of bring them in to try it. And they're all kind of a little bit skeptical, a little bit afraid and confused, but then they kind of try it and say, well, this doesn't taste like meat.
Yeah, maybe some people might be quite excited by the idea of trying novel foods, but yeah, it's all about trying to gauge their actual response, their physical biological responses, and not just hear their what they think is that how they think they feel about it, because for example, if I was asked, if I was told about the horrors of eating too much chocolate, um, and then was asked, okay, so now you've learned all of this, will you cut down on the amount of chocolate that you eat? I'd be like, yeah, of course, but no, it's not going to happen. I know for a fact that I'm actually, but my intentions are there. But would I actually cut down on my chocolate intake? Probably not. So that it's the same thing. Sometimes when you take a survey, you don't really get the real honest response. So this is trying to
Yeah.
mitigate that.
Perhaps you should speak to Caroline Cotto from Nectar. Have you heard of her? I mean,
No.
they do kind of taste uh, acceptance studies of consumers um, for plant-based foods. So they're kind of working with quite a few plant-based food companies to sort of
Okay.
see if people actually like it or not,
Yeah.
before it goes to market.
Thank you.
Yeah. So you co-founded Rooted Research Collective. So what is their purpose, and how does that kind of complement your academic research?
Rooted is an organisation that I started with two of my best friends, Marlana and my sister Lucy. So we basically focus on making sense of our complex, messy global food systems. So we design and deliver research projects that help accelerate a transition towards food systems that are more ethical, sustainable, and better for everyone. So one of our most recent projects was on the spread of dietary misinformation online. So we looked at the methods of spread, tactics, motivations behind some of the misinformation super spreaders, and we also recently ran a 6-month course for young journalists, and this was to help. This was in collaboration with Green Queen Media, and we helped shape how journalists talk about the climate and animal agriculture. So we're also working on consumer trust, education programs, culinary workshops, um, and yeah, so where my PhD is focused on the technology behind cultivated meats and
Mm.
alternative proteins, Rooted focuses on the human side. So my physical science informs the social research, and then the social research helps ensure that the science works better for society.
Yeah, the sort of misinformation and disinformation, the super spreaders, as you refer to them, that because there was research done by the Changing Market Foundation,
Changing
I think
markets, yeah.
it's yeah. I interviewed Nusa on a podcast, and she said that when in their research they found that even though there's over 150,000 clicks or something, it was only 20 people who were actually kind of spreading this stuff on, through Twitter and kind of various other
Yeah,
social media.
there's a handful of super spreaders. So when we analysed over 1 million posts, social media posts on Instagram, and we found that the majority of posts were by 24 super spreaders. 24 super spreaders. Oh, it was a while ago now. I'm hoping I've
That sounds
that
about
right.
what she was saying. Yes. Yeah.
Yeah, so these people, I might have got that number wrong, but Mar and Lucy are going to be very annoyed at me. But yeah, so they reach 24 million people. This handful of super spreaders, they reached
24 million people.
24 million people. I know that one's correct. So they had a widespread reach. And yeah, it was interesting to see that pretty much all of them had an obvious financial motive.
Because they have a cattle ranch or something, or because they're kind of.
Yeah, it was usually things like they had a consultancy service or they sold their own supplements. They all had a product or a service to sell.
I mean, it's amazing that I suppose it's the idea that kind of bad news travels faster than good news, right? So if they kind of say the awful things that can happen with plant-based food, like I saw, there were things in the Telegraph saying implying that plant-based food gives you cancer and things like
Mm,
that. So
mhm.
so clearly, I mean, I've written about that as well, and also at the conference in Chicago, I'm putting together and inviting speakers for that. There is going to be a session on misinformation and disinformation. So hopefully, some of the companies like Cargill can be can actually, I'm sure they'll refuse to even engage, but we shall see. But it's a big part of the food systems change, isn't it? Obviously, trying to
Mm.
inform people
Hmm.
properly. So, what did you find that were the messages that were spread so easily through your research? These kind of negative messaging.
Well, it was things that would speak to people's real fears and frustrations. What the super spreaders did really well was that they appealed to people's problems, and then they used emotion and storytelling to persuade them that this simple solution could solve all of their ailments or all of their issues. But I think we're all prone to misinformation. I think it's quite easy for us to go, oh, well, this changed my life, so it could change yours. But it was just that it was the confidence with which they spoke, the fact that it was a simple silver bullet that they were offering, and it was often portrayed alongside pictures of them looking incredibly healthy, incredibly fit. They were living the best lives. They're traveling across the world, and of course, because they're earning lots of money from their social media accounts. There was one person we looked at who was, it was estimated that they were earning at least $100,000 a month just through their social media platforms. So yeah, but it's storytelling really that gets people. And I think because life is so complex and fast moving and nuanced, that people want a simple answer to their issues. Um, it's it's easy to get caught up in okay, if you make this one change in your diet, it's going to have all these benefits, rather than having to go, okay, no, actually, I need to think about my nutrition. I need to think about how much sleep I'm getting. I need to think about this, that. It's too much nuance. People want a simple solution to things,
Yeah, for sure.
understandably.
Yes, and and clearly there are things that they're saying that are incorrect, right? I mean, so what are some of the sort of misconceptions that you kind of most think need correcting that perhaps some scientists can communicate more effectively themselves?
Well, if we're thinking about cultivated meat, or to be honest, any um, food systems related topic, I'd say the hottest topic at the moment is UPF. So that's ultra processed foods. Um, Many people instinctively lump cultivated meats into that category, and pretty much all alternative proteins, so plant based as well. So the first misconception that I'd love to correct is that these are that UPF automatically means unhealthy. It doesn't. So there's much more nuance. And I'd really recommend checking out the work of Jenny Chapman and Amy Williams on this if you'd like to learn more about UPF. The second misconception is that cultivated meat is some strange artificial creation. In reality, the animal cells are doing exactly what they've always done. They're growing, but they're just doing this in a clean and controlled environment that avoids antibiotics and disease and contamination and slaughter, and environmental damage. Um, we've actually been harnessing cells to transform food for thousands of years. So kombucha is made by a culture of bacteria and yeast. Um, Miso, soy sauce, cheese, kimchi, sauerkraut, yogurt. These are all processed foods, but they're ancient and they're accepted. So growing stem cells isn't new either, to be honest. It's something we've done for decades now for the tissue engineering industry. And cultivated meat, it feels unfamiliar because it's new to the public conversation, but it is something that we've been working on for a while. Um, but yeah, as I've mentioned, misinformation sticks because it appeals to people's emotions. And once the negative narratives take hold, we're left trying to play catch up.
Right. Brilliant. One last question before I let you go, Alice. So looking ahead, sort of 5 to 10 years, where do you think where do you think we're going to be with cultivated meats, specifically using your your particular technology advances?
Well, obviously my scaffolds are perfect, so no further breakthroughs needed there. But no, joking aside, I think the biggest advances that we need um, are probably on the cell side of things. So we're still looking for a stem cell that um, grows really quickly, that's high quality, and that is immortalized. So what that means is that it can multiply indefinitely, um, and one that can reliably develop into muscle and fat. Um, so I actually think this is somewhere that we could collaborate with farmers. So I didn't mention earlier, I forgot to answer that part of the question, Alex, when you said, how can we improve our communication? And I really believe that inclusion is so important here. If we invite people into the process early on, then they're going to feel more like they're part of the solution, that they've helped to shape it and develop it according to their needs, rather than just having something imposed from above. And this could help us to mitigate any issues that we have surrounding uh, things like farming. I know that there's an anti-farming sentiment. Um, and if we work with farmers on trying to create the best cell lines, then we might be able to improve consumer trust, and also make sure that the technology is going to improve the lives of everyone and not just the elite. Um, Farmers have this like deep knowledge of genetics and breeds, and how we can grow animals in different environments, and which animals cope best in like hot climates. And so yeah, I really do think that we could work together with farmers uh, to create the best cells.
So do you think farms could have bioreactors, um, which they would use to grow meat on their farms? Would there be like a like Respect Farms are trying to do in Holland with Ira van Heelen? So
Mm.
do you see something like that happening more widely, perhaps in the UK, where farmers and scientists are working together?
Yeah, I really hope so. I'm going to draw on my experience with fungi and my love of fungi again to tell you a story about how amateur scientists and community scientists have helped to contribute to developing a technology. So there was a time when we used to grow fungi only in labs, because you have to create this sterile environment for them to spread through their substrate. And if you tried to do that at home, there would be contamination, and the contamination would outcompete the fungi. Until this amateur mycologist, um, he discovered that if you sterilize the substrate inside a bag, and then you just add this little injection port to inoculate it, to add the spores of the
Mm.
fungi, you could keep the environment sterile. And this allowed people to start growing mushrooms everywhere. You don't need a big fancy lab to start growing them. So I really think that these sorts of developments, and working with food distributors and chefs and farmers, will enable us to try and scale out the technology a bit more, and we can work on finding new ways to produce cultivated meat that don't require a big fancy lab or manufacturing facility.
So do you think there might come a point in time where people could grow their own meat at home?
I like to think so. Yeah, there's actually a company in Japan that are already doing this. They're using things like a pressure cooker to sterilize and to create a cell culture environment. Is it Shojin Meats? I can't remember the name.
I've heard of
Um,
shitting meats. Yeah, yeah,
yeah,
not sure what.
yes. They're excuse me. They're already starting to create cultivated meat in home environments and at schools as well. So I think it's entirely possible.
That's very interesting. Yeah. No, I think that's a good note to finish on. So, Thanks, Alice, for coming on Future Foods interviews, and I will be watching with great interest your work and have a great Christmas, and thanks for coming on the show.
Brilliant. Thanks for having me, Alex.